7 decades since the Dead Sea Scrolls were discovered, around 80 new fragments of the ancient texts have been revealed to the public.
To serve our most important document with the technology that it was developed for our most important historical document.
70 years. That's how long the world's best biblical minds studied the Dead Sea Scrolls and reached the wrong conclusion. Not a small error, a fundamental misunderstanding of what the scrolls actually are, where they came from, and what they tell us about the origins of the Bible itself. The theory was so convincing, so carefully built, that virtually no one questioned it. Until a geneticist with no background in biblical studies sat down for lunch with a scholar in 2012 and asked one question that nobody had thought to ask.
As telling and as informative as the content of the text. And this has changed dramatically my own personal understanding of the Dead Sea Scrolls.
The answer came back in 2 words, and those 2 words unraveled 70 years of certainty in a single afternoon. The discovery that changed everything. In 1947, a Bedouin shepherd boy threw a rock into a cave near the Dead Sea and heard something shatter. When he climbed inside, he found ancient clay jars containing leather scrolls wrapped in linen.
Back the next day, climbed into the cave, and what did they find? They found huge clay jars.
Those scrolls became the most significant archaeological discovery of the 20th century, the Dead Sea Scrolls, the oldest known biblical manuscripts in existence, dating back over 2,000 years. For 7 decades, scholars poured their lives into these texts. They analyzed handwriting styles, compared linguistic features, and examined the physical appearance of the parchment. They used every tool available to traditional archaeology and textual scholarship to understand where these precious manuscripts came from and what they meant.
Pieces of the puzzle, and we can add them to our greater picture of the period and of the text.
The consensus that emerged shaped how the world interpreted the texts, how scholars understood early Judaism, and how historians viewed the birth of Christianity. It was a theory built on decades of careful work, supported by the best minds in biblical studies. It was also wrong. 25,000 fragments and no map. The Dead Sea Scrolls consist of roughly 25,000 fragments, representing about 1,000 different manuscripts. They were discovered in 11 caves near Qumran between 1947 and 1956. The texts include the oldest known copies of biblical books, sectarian, religious writings, community rules, hymns, prayers, and commentaries. They date from approximately 250 BCE to 68 CE, spanning a crucial period in Jewish and early Christian history. When the scrolls were first discovered, they were remarkably well preserved, protected by the dry desert climate and sealed in clay jars. But 70 years of handling and study had taken their toll. The scrolls are now extremely fragile. Some fragments are so small and damaged that scholars can barely read them. And because the scrolls are considered sacred texts by multiple religious traditions, any destructive testing has been strictly forbidden. Here's the catch. The sheer scale of the puzzle was staggering. 25,000 fragments, most of them tiny pieces of parchment, with just a few words or partial lines visible. No titles, no page numbers, no clear way to tell which fragments belong to which manuscripts. Like trying to assemble 1,000 different jigsaw puzzles mixed together, with most of the pieces missing and no pictures to guide you. Scholars had to rely on educated guesses, handwriting analysis, parchment texture, textual connections, and a wrong match could distort scripture or ancient tradition. If you placed 2 fragments together that didn't belong, you could create false readings, attribute ideas to ancient authors that they never wrote, generate hybrid texts that never existed. The margin for error was enormous. The consequences were severe. If this is the kind of story that makes you see history differently, that science can walk into a 70-year-old debate and tear it open, hit subscribe, because what the DNA found next is going to completely change how you think about the Bible itself. The Essene theory, confident and wrong. The dominant theory that emerged from decades of painstaking work was that the scrolls belonged to the Essenes, a Jewish sect described by ancient historians Josephus and Pliny the Elder. According to this theory, the Essenes lived in a monastic community at Qumran, isolated from mainstream Jewish society. following strict purity laws and copying sacred texts. When the Roman army approached during the Jewish revolt in 68 CE, the Essene supposedly hid their library in the nearby caves, where it remained until 1947. This theory explained many features of the scrolls. The sectarian writings describing community rules fit with what ancient sources said about the Essenes. Liturgical texts showing a solar calendar different from Jerusalem's lunar calendar suggested a group that had broken from the temple establishment. Apocalyptic texts expressing hostility toward the Jerusalem priesthood matched the image of a radical separatist group waiting for divine intervention. For decades, this became the accepted explanation, taught in universities, presented in documentaries, described in museum exhibitions. The Dead Sea Scrolls were the library of the Essenes. Case closed, but there were problems. Problems that nagged at scholars even as the Essene theory became dominant. And get this, the scrolls showed significant textual variation. Different versions of biblical books that didn't match each other. Some fragments written in different scripts by different scribes. The parchment quality varied enormously, from fine, carefully prepared skins to rough, poorly processed materials. And radiocarbon dating suggested that some scrolls were older than the Qumran settlement itself. Most puzzling was the sheer diversity of the texts. If these were the private library of one isolated sect, why would they possess multiple conflicting versions of biblical books? Why preserve texts representing theological positions they disagreed with? These questions troubled scholars, but without hard evidence, the Essene hypothesis remained dominant. Here's the part nobody talks about. The theory wasn't just accepted, it was sealed. A generation of scholars built careers on it. Textbooks were written. Museums designed entire exhibitions around it. For 70 years, the consensus calcified into certainty. Then came the lunch that cracked it open. The conversation that started a revolution. It was 2012. Oded Rechavi and Noam Mizrahi were sitting across from each other at Tel Aviv University, talking about the scrolls, about the maddening challenge of matching 25,000 fragments without any objective method to prove which pieces belong together. Rechavi listened as Mizrahi described the frustration.
Very material, the biological material of which the scrolls are made, is as telling and as informative as the content of the text.
Scholars were making educated guesses, handwriting analysis, parchment texture, educated intuition, nothing definitive, nothing that couldn't be challenged. Rechavi, a molecular biologist whose career was built on DNA analysis, went quiet for a moment. Then he asked the question, "What are the scrolls made of?" Mizrahi replied, "Parchment, animal skin." Rechavi paused. Then the parchment has DNA. That single sentence changed everything. It was one of those moments where two completely different fields intersect and create something entirely new. Mizrahi understood the textual problems that had puzzled scholars for decades. Rechavi understood the biological solutions that might address those problems. Together, they realized they could use ancient DNA analysis, a technique that had already revolutionized archaeology by revealing human migration, patterns, and population history, to answer questions about the Dead Sea Scrolls that traditional methods couldn't resolve. But there was one enormous problem. The Dead Sea Scrolls are sacred texts revered by multiple religious traditions. They are incredibly fragile after 2,000 years of aging and 70 years of study. Any technique requiring samples to be cut from the scrolls, cores drilled, or material removed would be absolutely forbidden. The scrolls had to be preserved intact for future generations. How do you extract DNA from ancient manuscripts without touching them? The dust that held the secrets. The team that eventually formed around this idea included Rechavi, Mizrahi, and Penina Shore, curator of the Dead Sea Scrolls at the Israel Antiquities Authority. They spent years developing a methodology that could extract sufficient genetic material while respecting the absolute prohibition against damaging the scrolls. And this is where it gets strange. The answer was hiding in plain sight, in the dust. Over decades of handling and study, tiny particles of parchment had accumulated as dust in the containers where scroll fragments were stored. This dust contained collagen, the protein that makes up animal skin, and embedded within that collagen was ancient DNA. By carefully collecting this dust from blank margins or damaged areas where no text existed, they could gather enough material for genetic analysis without touching any written portion of the scrolls. It was non-invasive sampling at its most ingenious, using material that would otherwise be swept away to unlock secrets hidden for 2,000 years. But extracting usable DNA from ancient parchment presented extraordinary challenges. The parchment-making process itself, soaking, scraping, stretching, and drying animal skins, damages DNA at the molecular level. The skins were then written on with iron-based inks that are chemically corrosive, further degrading genetic material. Then they sat in desert caves for 2,000 years, exposed to temperature fluctuations, humidity changes, and mineral deposits. And then they were handled by countless people over 70 years of study, each person potentially leaving traces of modern DNA. Stay with this for a second. They needed to find genetic whispers inside parchment that had been chemically attacked, cave-aged, and touched by hundreds of human hands. Without removing a single fiber from the scrolls, the team had to work in specialized ancient DNA laboratories with the strictest contamination controls imaginable: full protective suits, positive, air pressure, ultraviolet sterilization between every use. They focused on mitochondrial DNA, more abundant in ancient samples because each cell contains hundreds of mitochondria, giving them enough material despite the degradation. They developed strict authentication protocols to distinguish genuine ancient sequences from modern contamination, looking for specific damage patterns that only occur over centuries. Years of work, hundreds of samples, thousands of hours, and then the results came in. What the DNA revealed next didn't just answer one question, it detonated the entire 70-year framework. Cows in the desert, the first bombshell. The first major discovery from the DNA analysis seems simple on the surface. When the team sequenced genetic material from different scroll fragments, they found that most fragments were made from sheepskin. Sheep were common in ancient Judea, and sheepskin was the standard material for making parchment. That made sense, but some fragments were made from cowhide. That single finding broke everything open. Rechavi and Mizrahi understood immediately what they were looking at. Qumran, where the scrolls were found, sits in the Judean desert near the Dead Sea, one of the harshest, driest environments in the entire region. Rock, sand, sparse vegetation. Water is scarce. Grazing land is minimal. Sheep and goats can survive in this environment. They are adapted to desert conditions, requiring relatively little water and able to graze on the tough, sparse plants of arid regions. Cattle cannot. Cows need abundant water, dozens of gallons per day per animal. They need substantial grazing land with grass and vegetation. They are not desert animals. There was absolutely no way a community living at Qumran could have raised cattle. They could not have had regular access to cowhides for making parchment. The environment simply could not support it. That meant any scroll fragment written on cow parchment came from somewhere else. Somewhere with water and pasture. Somewhere outside the desert. Somewhere that could not be Qumran. The genetic evidence proved beyond any possibility of doubt that not all the Dead Sea Scrolls originated at Qumran, not some of them, not a few outliers. A substantial portion of the collection, some of its most theologically significant texts, had been written somewhere else entirely. And if the scrolls didn't all come from one place, the question of where they came from, what that collection actually represented, and what it said about the Bible itself, that question was suddenly completely open. Genetic fingerprints rewrite the Bible, but the DNA revealed far more than just species identification. By analyzing genetic relationships between different parchment samples, the team could determine whether multiple fragments came from the same individual animal or from closely related animals. This created a genetic fingerprint for matching fragments that might belong to the same manuscript. For the first time, scholars had an objective scientific method for determining whether fragments belonged together, instead of relying on subjective handwriting analysis or educated guesses about parchment appearance. Here's the part that made Mizrahi stop cold. The findings didn't just solve a puzzle, they forced a completely different reading of texts scholars thought they already understood. One of the most significant findings involved the book of Jeremiah. The Dead Sea Scrolls preserve two dramatically different versions of this biblical book. One version is similar to the Hebrew Bible we know today, longer, with chapters in a familiar order. The other is significantly shorter, missing entire sections, with chapters arranged in a completely different sequence. Scholars had debated for decades whether these represented genuinely different textual traditions from different communities, or just variant copies from the same tradition. The DNA gave a definitive answer, and nobody saw it coming. Fragments of the shorter version of Jeremiah were written on cow parchment. Fragments of the longer version were written on sheepskin. Two different animals, almost certainly two different regions, two different communities, each preserving their own version of the same sacred book, independently, in parallel. The differences weren't scribal errors. They weren't copying variations. They were two distinct Jeremiahs, each with its own geographic and theological home. Another crucial test involved the Songs of the Sabbath Sacrifice, a liturgical text found both in the Qumran caves and at Masada, the fortress where Jewish rebels made their last stand against Rome in 73 CE. Did the Masada text come from Qumran originally? Were all copies derived from one source? The DNA analysis showed that multiple fragments of this text found at Qumran came from different individual animals, separate copies, not one scroll broken apart. That multiple independent copies. The text was being reproduced by different communities across the region, not guarded by one sect as its exclusive property. And get this, the kinship analysis went even deeper. By examining nuclear DNA markers, the team could sometimes determine that parchments came from animals that were biologically related, siblings sharing significant portions of nuclear DNA, or parent and offspring showing characteristic inheritance patterns. When the researchers found clusters of parchments made from closely related animals, it meant those parchments were produced in the same location at roughly the same time. What the team discovered was exactly what you would expect if the scrolls came from multiple production centers rather than one isolated community. Some clusters of related parchments, batches made together in one location, mixed with completely unrelated parchments from entirely different sources. The genetic diversity was undeniable. Collection was assembled from multiple origins. It was not the library of a single community copying texts for its own use, a snapshot of an entire world. Stay with this for a second, because this is where the implications get enormous. For the book of Isaiah, different copies were written on parchments from animals with no genetic relationship. Multiple communities were copying Isaiah independently. The textual variance between those copies suggested different scribal traditions, different approaches, possibly different theological emphases in different places. If the scrolls were the library of a single isolated sect, the textual variations would be nearly impossible to explain. Why would one community preserve multiple conflicting versions of sacred texts? Why keep copies representing theological positions they disagreed with? It would be like a modern religious community maintaining multiple contradictory versions of their own scripture, possible but deeply strange. But if the scrolls came from multiple communities across ancient Judea, the diversity makes perfect sense. The collection isn't a window into one group's theology. It's a snapshot of religious debate and diversity across all of Second Temple Judaism. Different communities, different versions of biblical books, different approaches to religious law, different scribal traditions, all of it preserved together in those caves. The cow parchment fragments prove something even more specific. Texts were being physically moved between different regions. Someone carried scrolls written on cow parchment from wetter, more fertile areas into the Judean desert, where they were eventually deposited in the caves. That's not a sect hoarding its own library. That's a network. Communication, exchange, possibly pilgrimage between different Jewish communities in the period before the Roman destruction of Jerusalem. The DNA evidence supported a theory that had been proposed by some scholars but lacked definitive proof that the caves near Qumran served as a geniza, a sacred storage place where Jews from various communities deposited old, worn out, or theologically problematic texts that contain the divine name and therefore could not be destroyed according to Jewish law. Jerusalem is only about 20 miles from Qumran. When the Roman army besieged the city between 68 and 70 CE, refugees fleeing the destruction might have brought sacred texts to the caves for safekeeping, adding them to materials already stored there over previous decades or centuries. That would explain everything: the diversity, the multiple textual traditions, the texts that were demonstrably older than the Qumran settlement. the mixture of sectarian writings alongside mainstream Jewish texts that any community might use. Not one sect's hidden library. The accumulated religious literature of an entire civilization, preserved together in the desperate final hours before Rome burned Jerusalem to the ground. What this means for the Bible. Here's what nobody expected when Rachavi asked that question over lunch in 2012. The DNA didn't just solve an archaeological puzzle, it fundamentally changed what the Bible is. The DNA study demonstrated that the biblical text was not monolithic or uniform in the Second Temple period. Different communities used different versions. There was not one universally acknowledged correct text that everyone followed. The textual tradition was fluid, diverse, and still evolving. The process of canonization, deciding which version of a text was authoritative, was still ongoing and unresolved when these scrolls were copied. 2 different Jeremiahs, multiple Isaiahs, liturgical texts circulating independently across dozens of communities. The Bible, in the centuries before Rome destroyed the temple, was not a fixed object. It was a living conversation happening across an entire civilization. This has profound implications for understanding both Judaism and Christianity. The diversity preserved in the Dead Sea Scrolls shows the theological ferment of the period when Christianity emerged from Judaism. The debates over scripture, law, messianic expectation, purity regulations, and community organization reflected in the scrolls provide crucial context for understanding the world in which Jesus lived and in which the early Christian movement developed. Researchers are already pursuing the next wave of questions, analyzing DNA from more fragments, combining genetic data with multispectral imaging that can read faded or damaged text, building comprehensive databases of ancient animal DNA from different regions, hoping to create geographic reference maps that might pinpoint exactly where specific parchments originated. The work continues, but the verdict on 70 years of consensus is already in. Here is exactly what was wrong. The scrolls were not the private library of the Essenes at Qumran. They were not written by one sect in one place, and hidden in those caves during the Roman invasion. That theory, built by brilliant scholars over 7 decades, taught in universities, displayed in museums, printed in textbooks, was built on an assumption that no one had thought to challenge with biology. The assumption that all the scrolls came from the same place. They didn't. Here is exactly what the DNA proved. The collection came from multiple communities across ancient Judea, multiple scribal traditions, multiple versions of sacred texts preserved independently in different geographic locations, brought together in those caves from sources that range from the Judean desert to the wetter, fertile regions where cattle were raised. Not a sect's library, a civilization's archive. Oded Rachavi asked what the scrolls were made of. Noam Mizrahi said animal skin. And two words that every scholar for 70 years had walked past without stopping. Unlocked a completely different history of the Bible. The story wasn't written in the text. It was written in the DNA of the animals whose skin preserved those words for 2,000 years. And it waited there, patient and invisible, until someone finally thought to look. If you want to understand how another ancient mystery was rewritten by modern science, watch this next.